purification, as well as the finalization of the downstream processes. And we are looking forward to great presentations from Prof. Song, as well as Prof. Hun Jong, or from Kenai Beta Institute as well. So to start with, I just want to give a recap of what we got yesterday from our colleagues. Recap. All right. Here you are. I think the content that we received yesterday was very great, and so much content. And Julia, today, let's make sure that we send the presentations to the participants so that they can also have time to meditate upon them. But what I could take home, I think there were a lot of take homes, but for the sake of time, I'll just summarize the key issues. Was just the elaboration of the process flow for vaccine development and manufacturing, which we see many times during the GMP inspections, which they start with the vaccine candidates, and then the expressions and the creation of the stable lines, and then the upstream processes, which culminates in the formation of the cell bankings, and then the downstream processes and formulation, as well as fill and finish. And this was clearly articulated in several slides, as well as by both the presenters from yesterday. Then the whole process of how we actually get the master cell bank, as well as the working cell banks, when we go on site, it starts from the transfection and the host cell, then selection, and the master cell, when it's selected, it is then expanded, and we create several vials of these master cell cultures, and then these are frozen into the working cell banks that we see at site. And the working cell, the master cell banks that we see, and then the master cell bank vial is then, each vial is expanded into several cultures, and these cultures are then frozen into working cell banks. So we then see the working cell banks that are then used in the downstream processes. So another take home that I also took yesterday was quite an interesting illustration of the manufacturing process of the viral vectors through triple transfection and cell cultivation, where we see these working cell banks. If you see from the very first picture, this is what we actually see when we go for inspection. These working cell banks are then expanded in shake flasks, and then expanded also in the bioreactors. Before I used to think that a bioreactor is a very huge piece of equipment, but these are not so huge.
Recording in progress.
Them in shake flasks, then the expanded mixture is then put in wave bioreactors in the upstream processes with the cell growth, transfection, and lysis. Then you come to downstream processes where there is lysis clarification through depth filtration, and then you go to first concentration via diafiltration or TFF. And then you go to the chromatographic phase, the first capture as well as the second chromatographic, which is polishing. And then from that, you go to the second concentrate diafiltration, the diafiltration, the TFF. And then from that second TFF, you go to the final sterile filtration. aseptic filling. But what also caught my interest yesterday's presentations was the aspect of expression host. We have so many times seen the reference to the mammalian cell or the CHO, the Chinese hamster ovarian cell. So yesterday it was quite clarified by the presenters that the mammalian cells, particularly the human cell, is the ideal host cell platform to produce human proteins. And the commonly used types is this CHO, which is the Chinese hamster ovary cell, as well as the HEK293 cells. So there are several advantages of these host cells, and some of them, including E. coli, yeast, insect cells, have their own pros and cons. So basically, there was quite a lot of in-depth discussions on the upstream processes from both presenters from yesterday. I would like to believe that today's presentation is picking up from yesterday, and I want to then go back to the agenda of the day. Just bear with me. Unfortunately, I couldn't put up the slide that I was that I had prepared. But going back to the agenda, today we are going to do first the purification from Prof. Song. And I'll just give a brief introduction of Prof. Song. Before I go on to introduce Prof. Song, let me just acknowledge and introduce the presence of some of our leaders from the GAPTC who are joining us today. I see Don Mangwana, I see Mari, I see Dominic. I don't know if there are other GMPTC members, but if you are there, we fully acknowledge your presence and we thank you for joining us today. Yeah, I think I could see Don, Mari, as well as Dominic. I don't know if they are able to speak, but Don, are you able to speak? Julia, can you make Don and Mari speak so that they can meet the team?
Well, thank you very much, Washington, and good morning, colleagues. Glad to be part of the training. I've been attending, I think, since Monday, and there's a lot that I have learned as well. And I'm looking forward to today's sessions as well. And I just want to wish everybody a good session. Good morning.
Thank you very much, Don. Mari, you couldn't say something yesterday. Are you able to speak today, Mari?
Bonjour, Washington.
Yes, go ahead. Yes.
Bonjour, vous m'entendez? Bonjour, vous m'entendez? Je suis Marie-Laure Yété, membre du GMPTC de la République centrafricaine. Washington, vous m'entendez?
Yeah. All right. Thank you, Mari. Yes.
OK.
All right.
Donc, je suis Marie-Laure Yété.
All right. No, thank you very much, Mari. for the introduction. So Mari, Don, and other colleagues are within the GMPTC, which is the overall guiding technical committee that is responsible also for planning and approving these training sessions. So thank you very much. I'll go back to introducing Prof. Dunasong. Prof. Dunasong, he has been a postdoctoral fellow at Yonsei University since in September 2020. Then 2021, he was a senior research scientist at S2C Bio. And then up to 2023 May, he has been professor of Department of Graduate Program of Industrial Pharmaceutical Science at Yonsei University. And then from 23 up to present, he has been a professor of cannabis at Yonsei University as well. And he holds a BSc in biotechnology from Yonsei, as well as a PhD in biotechnology at Yonsei University as well. So Prof. Song, we are delighted to have you today, and we are looking forward to a great session from your side. You can meet the African GMP inspectors. The podium is yours, say.
Thank you very much, so the excellent introduction. So, can you hear me?
Yes, we can hear you very well and clear.
Yeah, thank you. Thank you. Hello, everyone. My name is Tuna Song, and I'm in KNRBRT of Yonsei University. So it's a great honor to be able to speak here today. I'm very happy to be here. So today I will talk about vaccine manufacturing bioprocessing, focusing on purification. So let's move on. Okay. The main goal of purification is to remove other impurities and keep the desired product. It is important to maintain the product structure and effectiveness. As an aspect of the manufacturing bioprocessing, purification must achieve this goal while considering several factors. First, minimize the loss of the product. We don't want the product to be work hard to disappear. And second, minimize the sample handling at every stage. Reducing sample volume quickly can reduce the burden on the next processing step. And third, use a different technique at each step. Each purification technique has different specificity and selectivity. So by using a combination of the techniques, we can selectively remove different types of impurities at each step. This ensures that the final product is high purity. And using different technique helps eliminate contaminants more effectively. And lastly, minimize the number of the steps. From cost effectiveness, it is beneficial to reach the final product with fewer steps. Before we start the main, I just briefly talk about the goal of the purification. So in the manufacturing process, the main techniques for downstream processing are chromatography and filtration. So today I will talk about overall chromatography and column packing. And for filtration, I will explain thermal flow filtration and tangential flow filtration. And lastly, I will cover the tests used to check the integrity of these filters. So first, I will talk about chromatography techniques and column packing. So what is chromatography? The purpose of chromatography is to separate the components of the mixture using a stationary phase and mobile phase. In this picture, you can see small beads inside the column and with a liquid passing through them. The small beads are the stationary phase called resin, and the liquid is mobile phase called buffer. As the mixture or sample passed through the column, each component interacts differently with beads and the liquid that cause them to separate. Besides resin and buffer, these components are also needed to perform pharmacography. And buffer tank hold the mobile phase. And pump. The pump drives the mobile phase through the column. It ensures a consistent flow rate and pressure, which is crucial for maintaining reproducible separate conditions. In downstream process, chromatography system often use dual pump setup for precise control of flow rate. And next, column. The column is a tube usually made of glass, acryl, or steel that packed with stationary phase resin. The column is where the sample interacts with resin. As it flows through, it allows for the separation of the different molecules based on their properties. The resin can have various chemical characteristics to separate molecule by size, charge, hydrophobicity, or affinity. Next, the detector. The detector identifies and measures the separated components as they elute from the column. It provides real-time monitoring of the elution profile by detecting specific properties of the molecule, such as observance at a certain wavelengths and conductivity and pH. This is a typical chromatography operation cycle. Equilibration is the first step, where we prepare the column to receive the sample. We do this by running a white buffer to separate through the column to balance it. This ensures the column environment is stable and conductive for the separation process. And load. In this step, we load the sample onto the column. The sample contains the mixture we want to separate. A proper loading is crucial for achieving optimal separation of the components in the mixture. And next, wash. After loading the sample, we wash the column. This step helps to remove any unwanted components and while keeping our target product on the column. The wash buffer selectively removes impurities without disturbing the bound target molecule. And next, elute. During elution, we added a special buffer that helps to release the target product from the resin, collecting it pure form. This buffer changes the condition within the column to favor the release of the target molecule. And next, regeneration. This step regenerates the column and resin, preparing it for next run by cleaning it and restoring functionality. And it involves running specific solution through the column to remove any residues. And next, sanitization. With sanitization, sanitize the column to remove any remain impurities and ensure it is clean for future use. This step typically involving using antimicrobial agent to ensure the column is free from microbial contamination. And next, storage. Finally, we store the column in a suitable buffer to keep in good condition until the next use. And proper storage condition prevent the degradation of the resin, ensuring they are ready for the next cycle. And these steps are the cycle that ensure the chromatography process is efficient and column is maintained properly. Each step is crucial for achieving high quality separation and maintaining the chromatography column. There are two modes for chromatography cycle, bind and elute and flow-through. In bind and elute chromatography, the product of the interest initially bind to the resin during washing step. During washing step, only impurities flow out, and the product elute during elution step. And flow-through mode, the product of interest elute during the washing step, and impurities binding to the resin in this step, and subsequently elute during the elution step. Typically, the bind and elute mode is more commonly used. So, when we want to purify our desired product, we need to choose the type of chromatography based on product properties. These properties including size and shape, and charge and isoelectric point, and hydrophobicity, and aggregation, and affinity. You need to decide which is these properties to use for best separation. So now let's talk about the different type of chromatography. First is affinity chromatography. This method purified a biomolecule based on biological function or unique chemical structure. It relies on a reversible interaction between target protein and specific ligand attached to the chromatography matrix, like this. Affinity chromatography is often used for capturing specific molecule from a mixture, and it's particularly useful for isolating components from very crude mixtures. Typically, this method operates in a binding and elute mode. The target molecule binds to the resin and is then elute and released in the purified form. So, for example, to purify mRNA, use oligodeity resin, which is composed of oligodeoxythioimidine nucleotide immobilized on the resin matrix. This resin binds to the poly-A tail of mRNA molecules. Next, ion exchange chromatography. Ion exchange chromatography separates the molecules based on their charge. It works by using reversible interaction tool between charged molecule and ion exchange resin with the opposite charge. There are two types of ion exchange chromatography, cation exchange and anion exchange. In cation exchange, we used resin that is negatively charged. This type of resin binds to the molecules that have a net positive charge. In anion exchange charge, we use a resin that is positively charged. This type of resin binds to the molecule that have a net negative charge. mRNA or RNA typically carry a negative charge at pH 7. that allow the use of ion exchange chromatography for their separation. Next, hydrophobic interaction chromatography, for HIC for short. HIC is method used to separate components in a mixture based on how much they interact with hydrophobic ligands. This interact is usually enhanced by high-strength ion buffers. But very occasionally, HIC can be used to separate plasmid DNA from more hydrophobic cDNA, RNA, and even damaged form of plasma DNA using flow-through mode. But in this case, actually other techniques like ion exchange chromatography or affinity chromatography are more commonly used. HIC is generally used to separate proteins and other hydrophobic molecules. The three-dimensional structure of the protein is influenced by interaction with the molecule and with the surrounding solvent, like water. For protein, this in case solvent is water. As a result, hydrophobic side of chain are typically pushed to the interior of the protein to avoid the water. However, when the protein is in a high-intensity ion buffer, its hydrophobic region become exposed that allow this interaction to occur. So, I have explained the different type of chromatography. To use these types, you will need column that contain the specific resin. So column packing is the process of filling a chromatography with the stationary phase resin. So in this picture, the part that looks like milk is the resin slurry. Column packing involves filling the column with this resin slurry. Objective of column packing is create a homogeneous bed of the resin. So, homogeneous bed makes such things uniform flow. This bed, homogeneous bed, ensure that the flow is even throughout the column. And this homogeneous bed can get high resolution separation. Homogeneous bed make achieving high quality separation of the components. And then consistent performance. The column should perform the same way with every run. This means it create a reproducible bed. A reproducible bed ensure the separation characteristics such like resolution, efficiency, and capacity. These characteristics remain consistent from one batch to another batch. This is critical for obtaining reliable and repeatable results in the chromatography. However, heterogeneous bed can cause uneven flow path. This can lead to inconsistent results and loss of the product yield. Maybe we may lose some of our desired product because of the heterogeneous bed and reduced product purity. The final product may not be as pure as we need. So this left figure is optimally packed bed. This display high column efficiency. And this figure is poorly packed. This display low column efficiency. When the column packing is done right, we could ensure the robust, reliable chromatographic process, but when it is done wrong, you could risk expensive downtime and loss of the product. And additionally, even with homogeneous packing bed too densely can cause channeling and early breaking through. When the resin is too tight packed, the mobile phase cannot distribute evenly and tend to follow path of least resistance then create channels. And packed the bed too loosely, conversely, can create liquid gaps that make mixing occur. This can result in reduced resolution or decreased purities. So next, let's talk about the main packing method: gravity settling and flow packing and axial compression. First, to pack the column, we need to prepare the resin as a slurry in specific buffer or solution. So in this case, gravity settling, this method involves letting the resin settle down just by gravity. However, using gravity alone can result in uneven packing. The heavier beads settle at the bottom first, and lighter beads take longer to settle at the top. So to create a homogeneous bed, we need to apply force, like a flow and mechanical pact. Then we use force, the bed settling faster and more evenly. If we use flow to apply this force, it's called flow packing. If we use pressure to apply the force, it's called axial compression. In case of flow packing, first transfer slurry to the column using pump or suction. During this process, be careful not to damage the resin. Resin is very fragile. Once the slurry begins to settle by gravity and you see 2 to 3 centimeter layer of clear liquid on top, and next, secure the column with the top adapter. Then apply the downflow buffer to help pack the bed. Then lower the adapter to the correct bed height, like this. Axial compression similarly transfer the slurry to the column using pump or suction. After that, fill the adapter as before, and then start lowering the adapter at the speed faster than simple gravity settling. So finally, lower the adapter to the height of the bed. So now we've discussed the main packing method very simply, but it is important to note the actual packing a column involves many considerations and precautions. There are numerous factors to take into account and specific equipment to handle carefully. So this makes column packing a little bit complex and challenging task. Okay, after packing column, we need to check if it is properly packed for effective separation or not. In well-packed column, the molecule we are interested in should stay in narrow and symmetrical band of the liquid. We can measure this using two-factor HETP, height equivalent to the theoretical plate, and asymmetric factor. These factors may not directly predict the quality or yield of the purified components, but they can be used to assess the suitability of the column and system before starting the purification process. Before we look at these factors, let's first understand the entropy of the peak. Retention time or volume help identify the compounds based on the time it takes to pass through the column. And height of the peak indicate the concentration of the compound at its highest point. And area reflects the total quantity of compound in the sample. And with this, with half height indicate the sharpness and resolution of the peak. Narrower peak are preferable for accurate separation. So HETP is the height of the section of the chromatography column that theoretically represent one separation step or one separation plate. Of the mixture. HETP is calculated by dividing the length of the column by number of theoretical plates. And so imagine the column is divided into small fragments. Each plate is each acting as mini separator. So more plates, the better the separation. The number of the theoretical plates in a column can calculate using this following formula. 5.54 is just constant, and VR is volume or retention time elute from the sample application to peak maximum, and WR is peak width, a measure as the width of the peak at the half of the peak height, 50%. At this point, V and W need to be in the same unit, retention time or volume. And industrial requirement is that HETP value must be within 2 to 3 times the average bit size. The asymmetric factor is parameter used in chromatography to describe the shape of the peak in the chromatogram. It also provides insight onto performance of the chromatography column and the quality of the separation process. The asymmetric factor is calculated using following formula. A is distance from peak maximum and to the front side at the 10% of the peak height. And B is the distance from peak maximum to the back side. When the asymmetry factor is 1, the peak is perfectly symmetric. This is ideal. But when the asymmetry factor is greater than 1, the peak tails to the right. And when the asymmetry factor is less than 1, the peak front to the left, like this. Industry standard is 0.28 to 1.5. So One of the critical purification technique is filtration. Essentially, it involves passing a particle containing fluid through a filter membrane. Looking inside the membrane, you can see that it contains pores. Membrane containing different pore size are available for different application. The particle larger than pore size of the filter are retained by the filter, and particles smaller than pore size of the filter pass through the filter along with the liquid. Filtration can be classified in the following way. First, based on the operation method, there are NFF, normal flow filtration, and TFF, tangential flow filtration. Normal flow filtration can be further divided based on its purpose. Depth filtration and bioburden reduction and straw filtration. And tangential flow filtration is used for ultrafiltration and diafiltration. Sometimes TFF used for microfiltration also. Let's take a closer look at each of these types. Depth filtration is a method used to remove particles and impurities from liquid. These depth filters are different from other types of filters because they do not have a specific pore size. Instead, they have a thick, porous structure made of many layers and complex structure, and it allows it to trap particles throughout its depths, not just on the surface. As the liquid flows through the filter, particles get trapped within the multiple layer, and this makes depth filter very effective at capturing a wide range of the particle size. So, they are used for clarification and prefiltration, and they remove particles via side exclusion and absorption, and possess a high capacity for particulate matter. Depth filtration is commonly used in process where there is a high load of impurities. It's great for removing larger particles and detect more precise filtration steps. But they show the limited precision. Because they do not have a specific pore size, depth filters are not as precise as other filtration, such as membrane filtration. They are less effective at removing very small particles or microorganisms. Next, bioburden reduction filter. Bioburden reduction filters are used to decrease the number of bacteria and other microorganisms in the process. Here are some key points. There are filters typically have a pore size of 0.45 or 0.2 micrometers. Manufacturer may test these filters are bacterial reduction, but they do not guarantee complete sterilization. They can be used alone to control bioburden in several places in the bioprocess, before the chromatography column, and before holding steps or storage, and during buffer preparation. The main goal of this filter is to minimize the bioburden during the process. There is no need to validate sterility, just to reduce the growth of the microorganisms. In combination with other controls, bioburden reduction filters can be sufficient for certain process steps. Next, talk about the sterilizing grade filter. A sterilizing grade filter can remove at least 10 million aluminium forming units per square centimeter. This ability is measured using called LV, which stands for log reduction value. The LV shows how well the filter can remove bacteria. The equation for LV is the log of the ratio bacteria input to output. For example, if 10 bacteria go into the filter and 1 comes out, LV is 1. If 100 bacteria go in and 1 comes out, the LV is 2. If 10 million bacteria go in and 1 comes out, the LV is 7. This is what makes it sterilizing grade filter. Sterilizing grade is based on how well the filter works, not just size of the pore. These types of filters, like depth filtration, bioburden reduction, and straw filtration, use a method called normal flow filtration, NFF. Normal flow filtration works in a way similar to making drip coffee. As shown in the picture, the feed go into the filter and the filtrate comes out in the same direction. So in this method, the particles collect on the upstream side of the filter. This can lead to fouling, clogging, or caking on the filter surface, like this. So normal flow filtration are usually not reused, just single use. Tangential flow filtration is different from normal flow filtration. In TFF, the feed flow into the system at a right angle to the direction of the filtrate or permeate, 90 degree. Here's how it works. The feed enter the system and flow along the surface on the membrane, and the filtrate passed through the membrane while the retentate flow out separately. This means there is one input feed and 2 output, filtrate and retentate. Because the feed move parallel to the membrane, TFF system could prevent molecules from building up on the surface of the membrane. This could cause self-cleaning. This self-cleaning can make filter reused for several batches. Using such TFF system, we can concentrate product through ultrafiltration and perform buffer exchange. I'll talk in more detail later. So the feed solution is pumped from the feed reservoir through the filtration module, TFF filter module. As the feed solution flow across the membrane, pressure is applied. A smaller molecule passes through the membrane to the permeate side or filtrate side, and a larger molecule are retained. Larger molecule retained and returned to the feed tank. Retentate continues to flow parallel to the membrane and recirculate back to the feed reservoir separately. And permeate is filtered out and collected in a separate container or wasted. Such continuous operation and recirculation of TFF are beneficial for large-scale operation. And there are many steps for TFF filtration operate, including setup, ultrafiltration, diafiltration process, and NWP check for monitoring and cleaning. Before you process any product on an ultrafiltration and diafiltration unit, you must first perform setup and equilibration. In the setup stage, the first step in the operation is installation. First, we need to install the TFF filter into a suitable housing or holder, and then connect it to the equipment. This process can vary depending on the types of equipment or filter type you are using. While installing, it's important to be careful to avoid any leaks or damage. Make sure everything is securely in place and properly connected. And next, sanitization. There is how we clean and reuse a membrane for another batch of the same product. In this step, rinse the membrane with water, remove any leftover protein, then circulate such as sodium hydroxide solution through the system for a specific time. Then again rinse the membrane with the water again to remove any remaining chemicals. This is a water flush. And next, we can check within integrity test. In the case of TFF, we also perform a filter integrity test to ensure the filter meets the manufacturer's specification and the setup is correct. I will explain filter integrity test in detail later on. And next is determine-based MWP. So, normalized water permeability and WP, it is important to maintain filter health. This is for ensuring consistency in membrane performance. And WP measure how easily water pass through the membrane. By checking NWP regularly, we can make sure the membrane is working consistently and efficiently. And second, monitoring membrane fouling. In UFDF process, membrane get clogged over time, even though this is TFF. So if NWP decrease, it means contaminants are building up on the membrane. Monitoring NWP helps us detect this early, so we can clean the membrane when we need. And next, determine membrane replacing time. By keeping track of NWP, we can figure out how long the membrane will last. If NWP drops below a certain level, it's a sign the membrane is no longer performing well and needs to be replaced. Water permeability can vary based on pressure across the membrane and the size of the membrane as temperature. So by understanding and monitoring NWP, we can ensure our filtration system are running smoothly and effectively. The next step is buffer flush, which is same as equilibration. The purpose of equilibration is to precondition the filters before introducing product. The membrane are equilibrate using buffer solution. This makes to ensure the protein is not exposed to very different pH or ion strength conditions. So after buffer flushing, now the filtration unit is ready to accept product. The product can be transferred into the unit either from a feed vessel or in line via filter unit from another stage of downstream process. For the ultrafiltration step, the product is concentrated to the target concentration. During the filtration process, unlike NFF, it is less prone to the clogging or fouling. Once the target protein concentration is reached, the diafiltration process can begin. While protein solution is recirculating, a new buffer solution is added to the feed. As the diafiltration proceeds, the new buffer solution feed replaces the original buffer. So ultrafiltration works like this. During process, the product, which is larger molecule, stay in the retentate, and impurity, which is small molecule, are filtered out into the permeate with the buffer. So as this process continues, the product can be concentrated by reducing its volume, like this. And diafiltration is used for buffer exchange. In other words, placing buffer that was used in previous processing step with a new buffer that is suitable for next processing step. It is also method by which we formulate our drug product. It involves adding the fresh buffer called diafiltration buffer to the old solution, simultaneously filtering out small impurities through the membrane. This process is repeat several times to achieve the desired level of purification. And diafiltration continues until the filtrate matches the diafiltration buffer in terms of pH and conductivity. So in the filter, impurities and buffers are removed, leading to the concentration in here, and new buffer flow into the feed tank that allow for buffer exchange. So when performing ultrafiltration, diafiltration, there are important parameters: cross-flow rate, and filtrate flux, and transmembrane pressure, TMP. These three parameters need to be well controlled for UFDF process to work effectively. By understanding and controlling these parameters, we can ensure a successful UFDF process. So let's take a closer look at each one. The cross-flow or feed flow rate is the speed at which a solution moves through the feed channel and across the membrane, like this. This flow creates a force that helps sweep away molecules that could block the membrane. However, if a cross-flow rate is too high, it maybe can cause higher turbulence and potential tear, which might damage the membrane. Also, the feed has less time to pass through the filter membrane, which can make process take longer. Next, filtrate flux. The speed at the filtrate pass through the membrane is called filtrate flow rate or permeate flow rate, but filtrate rate, flow rate is depend on the total membrane area. So it makes difficult to compare between systems of different size of area. So filtrate flux is filtrate flow rate adjusted for area of the membrane. This means how much filtrate move through a square meter of membrane in an hour. It is essential to measure of how efficiently the membrane is filtering the liquid. The measure filtrate flux in LMH, which is a liter per square meter per hour. And next, transmembrane pressure, TMP. This is pressure difference between feed side and the permeate side of the membrane. The TMP equation like this, calculate by taking average the feed pressure and the return tape pressure, which are the pressure on the top of the membrane, and subtracting the permeating pressure, which is the pressure below the membrane. So why is TMP important in TFF? TMP is the driving force that pushes the liquid through the membrane, making the filtrate process work. And TMP performance indicator. By monitoring TMP, we can understand how well the membrane is performing. A stable TMP means the filtration process is consistent and working properly. And TMP can fouling detect. If TMP increases over time, it can indicate the membrane is getting foul and cakey. This means contaminants are blocked the membrane pores, which can reduce efficiency. So, understanding monitoring TMP is crucial for ensuring the TFF process runs smoothly and effectively. Such TMP varies depending on the membrane pore size and process conditions. Higher TMP can enable faster flow through the membrane, but can also increase the risk of the fouling. It's important to balance a TMP to achieve optimal feed flow rate while minimizing membrane fouling. So usually a small to moderate TMP is maintained to reduce the risk of fouling. The priority is to reduce fouling to keep the filtration process efficient. So after complete UFDF, it's time to recover the product. To recover the remaining product in the system, that is not just in the tank. We need to rinse with the buffer. Running buffer through the system that help because there are considerable amount of the solution held up in the system. And once processing product is finished, the unit must be thoroughly cleaned. For cleaning, sodium hydroxide is usually used to remove remaining components and buffer residues. In this case, UFDF assembly could be cleaned using the same cleaning protocol that was performed before use. Next, we rinse the unit and membrane with water to remove sodium hydroxide. After rinsing, we test NWP again to make sure the cleaning was effective. After using the membrane, we compare the permeability, how easily water can pass through it after cleaning with its pre-use value. This equation helps us see how effective the cleaning process was. By looking at this trend, percent membrane recovery trend, we can estimate how long the membrane will last and decide how many times we can use it before we need to replace. And different companies set their own standard for membrane recovery, usually range from 80% to 120% membrane recovery. As the measuring post NWP, we flush it with buffer to prepare for the next use. But if the filter are not going to be reused right away, they can be stored for a long time. To do this, we need to circulate an appropriate solution through them. Make sure to remove the residue and keep them wet during storage. The solution you use should be antimicrobial to prevent any contamination. Next is a final chapter, the filter integrity test. So, what does the membrane integrity? Membrane integrity means the filter membrane can do its job correctly. If the membrane is not intact, impurities and large particles should be filtered out, and it will pass through downstream. This is something we definitely don't want to happen. So why perform filter integrity test? And first, check correct installation. We perform filter integrity test to make sure filter is installed correctly. This test helps detect any system leaks caused by issue with O-rings, gaskets, or fault seals before actual process. And second, confirm manufacturer's specification. This test confirms that filter meets the manufacturer's specification. It ensures we are using the correct pore size filter for our process. And third, check for damages. We need to check the filter for any damages. This assures the filter's integrity before sterilization and after sterilization, like steaming or autoclaving, and or during transport. And fourth, regulatory requirements. The filter integrity test is also important for meeting regulatory requirements. It creates link between validation and current processing conditions. It is important for GMP and audit. So filter integrity testing is crucial for ensuring proper installation and verifying the right pore size, right specification, and checking for damaging and meeting regulatory standards. It's a key step to ensure our processes are safe and effective. During various stages of the manufacturing process, multiple filtration steps are performed, but not all of these are sterile filtration. In the downstream process, filtration is done for purpose of bioburden reduction. By reducing bioburden, we can ensure the purity of the product. During this stage, a filter integrity test is conducted for risk analysis to ensure the filters are functioning correctly and maintaining the desired level of the purity. On the other hand, in the final filtration step, it is crucial to guarantee sterility, like LV7, because this stage is subject to regulatory audit. Obviously, because this step is right before the final product. There are two categories of filter integrity test: destructive and non-destructive test. A destructive test involves challenging the filter with bacteria to determine its ability to retain microorganisms. The destructive test is the best method to evaluate the sterilizing filter's capability to retain the bacteria. In this experiment, we intentionally grow the microorganism and pass them through a filter. Then we take the filtrate and put it through it analysis membrane filter, and we grow this membrane for 2 weeks. If we see an LV of 7 or more, it means this experiment is successful for sterility. Because the filter becomes contaminated during the test, this test is conducted using a representative filter, not pre-use or post-use. Next, non-destructive test. This method is crucial for ensuring the performance and the reliability of filters without damaging. The main types of non-destructive tests are diffusion test, bubble point test, and water intrusion test. Filters can be divided into 2 types based on the affinity with water: hydrophilic filter and hydrophobic filter. Hydrophilic filter love the water. When the water touches these filters, it spreads out over a large area. Application of the hydrophilic filter is a filtration of aqueous solution in downstream process and filtration of drug products. On the other hand, a hydrophobic filter hates the water. When the water touches these filters, it builds up onto droplets instead of spreading out, similar to how oil and water don't mix. Application of hydrophobic filter is vessel vent filter and gas filters. Hydrophobic filters can be tested using diffusion test, bubble point test. On the other hand, a hydrophobic filter can be tested with all three methods, including water intrusion test. So this method, diffusion test, uses the property of the gas to diffuse based on the concentration. When the gas is applied at the constant pressure from the upstreaming to a wetted filter membrane, this gas dissolved into the wet solution, and dissolved gas then exit onto the downstreaming side. This make and create a gas flow. If the flow rate is higher than the specific limit, it may indicate have an issue with the filter's integrity. Next is a bubble point test. This method uses the force that keeps the wetting solution in the filter's pore due to the surface tension. The main factor is surface tension. The small pore size need more force to break the surface tension. So at lower pressure, the solution stays trapped in the pore because of the surface tension. As the pressure increases, there comes a point where the trapped solution is suddenly pushed out. This point is bubble point because bubbles comes out at this time. When this pressure is higher than the filter specified limit, it ensures the pore size and integrity are maintained. If the filter isn't integral or not well wet, it will be high gas flow rate at low pressure. To do diffusion test and bubble point test, the filter need to be complete wet. In case of hydrophilic filter could be wetted using water. However, hydrophobic membrane hate the water, repel the water. So how do we handle this? How could we wet the membrane? We use a mixture alcohol and water to wet hydrophobic filter for this test. But these tests are usually done offline to avoid contamination risk. Because of the contamination risk, it's not possible to test the filter in place after steaming. So using water for wetting is more suitable than alcohol because of disposable cost and other facts and contamination risk. Therefore, the water intrusion test was developed. The water intrusion test relies on the fact that the membrane is hydrophobic, meaning it repels the water. But this method is tested that can only be done with hydrophobic filter. So what happens when you put a hydrophobic filter in the water and apply the pressure like this? The water filter in. The water can't pass through the water. Instead, the pressure makes the filter crumple or shrink like this. Take a look at this figure. This is the filter before test, normal filter. This is the filter after the test. It is crumpled. So what happened during test? As pressure is gradually applied, the filter start to deform and create a water flow. This happened because the crumpling filter allow more water to be outside the membrane. This make water flow. The water was filled in the housing goes down because of this. And once it stabilizes, the water flow almost stop like this. This test is to confirm that there is very little water flow at this point after stabilization. This is how we ensure the filter's integrity through the water intrusion test. So, thank you for all the listening. So, I have gone over time, I think. So, I suggest I answer the question during a Q&A session following the next session. Thank you once again for your attention. I'll see you shortly.
Thank you, Prof. Song. Had some great presentation. I think you took your time to deeply delve into the aspect of purification, right from the column, theoretical column aspects, filtration, diffiltration. We had not had that elaborate presentation on these concepts before, and we thank you for your patience in delivering these aspects. I see there are a few questions that we can tackle maybe in not more than 10 minutes, so that we move on to the next presentation. Let me just put on screen here the questions. So, Prof. Singh, maybe if you could try to address what you can from the questions. Can you see my screen?
Yes, I can.
Yeah. So the first question, should the reuse of TFF be validated?
And TFF, should the reuse of TFF be validated? Before we use, we have to check the MWP, because before use, already know the before batches, after MWP and trend. So we have to check. So MWP can validate that, and if the product is of pharmacopoeia standard, but HPLC chromatographic conditions are not the same as those in the pharmacopoeia, is the manufacturer required to validate the analytic method? Sorry, I didn't understand the question. I think about that maybe after next session Q&A time. And what is the importance of checking the integrity test post-use? After completing filtration process, of course, I know that it's regular requirement. If the after processing, maybe test will, if test fail, that means the process we can't be sure to the process is good condition. We can't be sure. So if the filter integrity test fail after use, after process, we have to check the filtrate or downstreaming side. There are our predicted results or not. If the filter integrity test is pass, we can ensure the process was good in condition. And next, with regard to the chromatography spotlight, I just wanted clarification as to whether the system is employed for the sampling only for direct separation of the feed from the processing unit, and just want to maybe in downstreaming process, just direct separation of the feed from the processing. But depends on the scale. The lab scale could be applied for the sampling, but large scale, direct separation of the feed from the processing. And any concern of the particle size of column? The particle size of column means particle size of resin, I think. That maybe supplier give you the particle size of the resin, and downstreaming process, what the critical process parameters that are consistently monitored and were affected by multiple factors and can changing from one cycle to another. This is I can't understand that this is what for chromatography or TFF or just downstreaming process. It may be difficult to say what for the question is. So next, how often do you check the filter integrity test? Usually, in downstreaming process, the filter, there is a guideline depending on the company's guideline. For bioburden reduction, maybe just for risk analysis. So sometimes, often or all the time, but some companies often or just first time or different from the company's guideline. But sterilization filtration, always, every time, before use and after use. And filter integrity testing, the responsibility of the manufacturer, the filter, our manufacturer of the drug product. Yes, filter integrity test specification, like a specific limit. Maybe suppliers give you the specification, so you can know that. And what criteria are used to choose the filtration method to use? This is so tricky question. What technique you use is so depend on your product and product properties. So that's I can't answer that. Sorry. And so many things. Okay. During filter validation, what are critical process parameters can faulty attribute we need to consider? Filter validation, I can see the first question. Okay. For validation, maybe pore size and capability of the some microbial or bacteria. It is depending on the what type of the column. It is depending on the column type, filter type. So what validate for that? Okay. Filter surface. Usually filter surface, you have to check before the use the filter, the surface membrane, filter's membrane interact with your protein. Have to check if there are some bind or interact, maybe you can lose your product. So you have to choose not interacted with the filter membrane. And what are the acceptable limit for the filter pressure drop and flow rate to ensure consistent performance? I think this is for TFF. Maybe this is also depending on the company's guideline. It depends on the product or process condition. So have to research and check before the manufacturing process. Can depth filtration replace centrifuge or the clarification step in our process? Sure. Filter depth filter and centrifuge or both are for clarification, but in these days, centrifuge maybe more make loss of the product, but in very large scale, centrifuge is preferred. But small scale or like 2,000 liter media, I think the smaller than 2,000 liter more prefer the filtration, use the filtration, and over the 2,000 liter, maybe centrifuge more preferred. And
Professor,
yes,
can I disturb you and maybe stop the Q&A at this time
yes,
so that
okay,
we do not approach into the next presentation? I know we're already 10 minutes into it, but
yes,
we will try to give some more time at the end of this presentation so that we can attend to all the questions.
okay, thank you.
All right. And I'll also give you this document so that maybe you can write down the responses and then we can share with the participants.
Yes, okay.
But otherwise, thank you very much for the presentation as well as the Q&A session. This was quite great and we are grateful.
Yeah, thank you for attention. Thank you.
All right. Our next presentation is coming from Prof. Won Jeong, who's also